So today my topic is interaction of the double helix with small molecules and ions. The interaction of the DNA double helix with the small molecules and ions is a significant area of study in biophysics, as it plays a crucial role in biological processes like replication, transcription, and repair. Binding of small molecules. Small molecules can bind to DNA through several mechanisms. The first is intercalation. Molecules like ethidium bromide, doxorubicin, and other planar aromatic compounds slide between base pair in the DNA helix. This intercalation distorts the DNA structure, which can impact processes like transcription and replication. Minor and major groove binding. Molecules such as netropsin, distemsin, and certain antibiotics bind with the minor or major groove of the DNA. The shape and hydrogen bonding pattern in these groups allow to selective binding, influencing DNA functions. Covalent bonding. Some drugs and toxins like cisplatin form covalent bonds with DNA base, often causing crosslink. This disrupts the double helix, leading to apoptosis, programmed cell death, and making these agents effective in cancer treatment. Now, electrostatic interaction with ions. DNA is highly negative charge due to its phosphate backbone, so it attracts counter ions to stabilize this charge. Monovalent bonds. Sodium and potassium ion shield the phosphate group negative charges, maintaining the DNA stability in solution. These ions also influence the helical structure and help maintain the B form of DNA. Divalent ions. Magnesium, calcium, and other divalent cations have a stronger stabilizing effect than monovalent ions and are essential for certain DNA processes. For example, magnesium is crucial in DNA polymerase activity during DNA replication and transcription. Multivalent ions and polyamines. Ions like spermidine and spermine can induce DNA compaction, folding it into a more condensed form. This is particularly important in cellular environment where DNA needs to be compacted into smaller spaces, such as in eukaryotic chromatin. Now, effects on DNA conformation and stability. Helical forms. The interaction with ions and small molecules can shift DNA between its various helical forms. For example, high salt conditions can favor the formation of Z-DNA, a left-handed helical form with the unique properties. Thermal stability. Binding of ions and intercalating agents can increase or decrease the melting temperature of DNA, temperature at which the strands separate. This is important for processes like PCR and understanding DNA stability under different conditions. Condensation and supercoiling. Polyamines and certain cations can induce DNA condensation with intercalators, and groove binder may influence DNA supercoiling, which is crucial for DNA packing in cells. Function implications. Gene regulation. Some small molecules can selectively bind to promoter regions of specific DNA sequence, influencing gene expression. The mechanism is used by transcription factors, which interact with DNA to either upregulate or downregulate gene activity. Anti-cancer and antiviral therapy. Many drugs are designed to bind to DNA and inhibit crucial cellular process. For instance, some cancer therapies rely on DNA targeting drugs that prevent replication or induce cell death by damaging. Spectroscopic and structural studies. Techniques like X-ray, NMR, and atomic force microscopy are used to study the interaction at high resolution, while circular dichroism, CD, and UV visible spectroscopy help assess structural changes in DNA induced by binding. Thank you.